just from here, the Department of Mathematics. So, so perhaps this is a slightly different talk to the rest. If you get lost, just stop me. We don't really have to go through all the slides. I'm hoping just to convey some messages. Okay, so as a statisticians start from data. I mean, it's the motivation from which we then develop some statistical model and some inferential procedure. So, as in fact, you will see two examples if we do get to the end. So the first one is on microbiome, this one here. So the data are microbial abundance, measured measured on some sample, biological sample, so typically quite a large number of microbes. In fact, in terms of data, they then get summarized into little clusters of microbes. So you will see in the next slides, OTUs, Operation Taxonomic Units. They are the random variables for us as statisticians that we would like to model. So one sample, so one patient, gives us this vector of abundances. And then the biologists do a number of experiments. In fact, also not just from the same location, sample taken from the same location, a number of them. This is data on that you will see on 13 body sites. I'm going to call them environments. So, for example, there's quite a lot of data in the mouth, so samples taken from saliva or from the tongue or from the throat, and so on. In fact, there's a lot of papers on saliva, for example, and they study the oral microbiome with that. There's quite a lot of papers taking data from stool and then studying the gut microbiome or the skin. So in this case, it would be samples from behind the ears and the nose. So these are different body sites. So what is it's essentially multivariate data. The random variables on the microbes, and we have those for different body sites. The general question of interest in the field, and then the one we are trying to answer is to, so there is interest to know how these microbes interact with each other, right? They keep the system functioning by a network of interactions, and these are unknown, and so we want to recover them from data. This is the general question. The field seems to think that there is a core network, so some interactions are always there, so some basic, somewhat functioning. So whether you are looking at the oral, the gut, or the skin, they are kind of there, some basic kind of building blocks. But some interactions may be specific to each environment. So they actually are related to the gut microbiome or the skin microbiome or the oral microbiome. That's quite important. So we would like to learn both this kind of core network, but also specific interactions. So these two are really interacting in the oral microbiome, these two in the gut microbiome, and so on. Okay, so just a quick exploratory data analysis, could say, of the data, just to give you an idea, because some of the peculiarities of the data will then need to be taken into account in the modeling. So it tends to be quite high dimensional. And in fact, it's 87 variables. It's not even that many. The data is 10,000. So there's some filtering. Some of them are all zeros, for example. But it tends to be quite high dimensional. This is the number of samples overall. So that divided by 13 is roughly the number of samples per body site. So on the left here, you see some box plot of the percentage of zero for each microbe, but I split it for environment. Generally, what these data are very sparse, so there is a large percentage of zeros, which does need to be taken into account in any kind of statistical model. And I should say it's count data. So the latest kind of biological experiments that they will give you data in the form of the number of biological fragments in a sample. And indeed, there are many zeros in here. So this depends a bit on the body side. For example, for the stool here, you see 99% as the median of zeros in the sample. So when you then do modeling, various people look at zero-inflated models, for example. The other thing is it's over-dispersed. So this you can see a little bit here. Just a quick exploratory check. So you look at the empirical variance divided by the empirical mean for each OTU. And if it's 1, it's kind of roughly Poisson. If it's less than 1, it's undispersed, and higher than 1 is over-dispersed. And it can be quite much higher than 1. So it's over-dispersed data. So in terms of modeling, people use, for example, negative binomial. It's a kind of distribution you can think about. And okay, this is a bit more kind of biological. So that for each experiment, the total number of fragments is can vary between a sample and another. So you need to somewhat normalize the different data for different samples. Okay, so this is the kind of setting and the data. So going back to the question, and in some sense here, I'm trying to connect with all of you. It's not what I'm an expert on, but it could be the closest to you. So because the question is indeed, we want to recover these interactions within each environment, but also similarities between environments at the level of a network. And so maybe some of you, I don't know, is there anyone working on tensor graphical models? Yeah. Yeah. So that could be indeed a type of model to consider. So just also to define the notation, yk is this vector, p-dimensional vector. P is the number of OTUs, the number of microbes. K is the condition, for us, the body site. So this yk is our random vector. We stack them all together. So we have a P times B vector, and we could assume, put in brackets, I put copula-Latin in the sense that the data are not Gaussian, but for what I'm saying now, it doesn't matter too much, but I mean, we should go beyond some kind of Gaussian assumption. But okay, let's now forget this for the moment. We could think of it tensor Gaussian graphical model. This, I believe, is one of the simplest ones. So this matrix here is a very large matrix. P times B. P was about 100. B was 13. So it's a large matrix. So we need to do some modeling, and the simplest one is this Kronecker product between the precision matrix related to the dependencies between microbes and the one dependencies on body sites. Is this useful? Well, it does answer a bit the question that we wanted to answer, in the sense that this P by P matrix here has something to do with dependencies between interactions between microbes. It doesn't quite answer what we wanted in some sense, at least with this simple model, where we don't have networks that are specific to each body site. So this is quite a simple and it's quite a strong assumption as well. So if you have a zero in this matrix between two microbes, they are not related, and they will not be related in any of the body sites, saliva, throat, and so on. I mean, we expect some similarities and some differences. So this seems to be quite strong. The other matrix could tell us something about, not directly, but something about relationships between body sites. Not directly, but I mean, if you see a positive association between two body sites, you would expect if the abundances are high for microbes in one body site, they will be high also in the other, and so the networks will also be related. So somewhat it's going in the direction of what we want to answer, but in a simple way. So there are, I mean, I don't want to enter too much into the models. Indeed, people work on this, maybe know of extensions, for example, to have microbe-specific networks, some kind of microbe effect. But the main limitation, as far as I can see, is that these models require data where a unit, a patient for us, is measured across all these conditions. Am I right? This is the main thing, the main point I would like to make. So you need to have you need to have the same patient where they make they do an experiment on the tongue, on the throat, on the stool, on the saliva, and so on. It's quite a rigid structure, particularly if you get to high-dimensional data. I mean, for example, longitudinal data, temporal data can be like this. You follow up over time, but in biology, it's quite unusual. So I hadn't looked at it before, but now thinking about tensor graphical models, I looked at the actual data. And for example, I noticed there is no single patient that has been measured for all the body sites. There is one patient that has measurement for 6 body sites, but that's one sample size one. You don't do much with it. So the most realistic thing you can do is probably this block here of 600 or so patients where you have measurements for 2 body sites. But perhaps I should check, maybe not the same one. So I mean, it's quite a specific structure that you require to use those tensor graphical models, and therefore we didn't even consider them, let's say, for these particular data. Okay, so there are other things that that are related to what I'm talking about that you may know. So we could, for example, do something like we call a joint graphical lasso. So again, there is this massive P times B times P times B matrix. So also here, in some sense, we do some assumptions. You use a block. You make it as a block diagonal matrix. So we assume a model for each condition. So the omega k now depend on k, and they are the networks related to the networks between microbes in condition k, body site k. So these are 13 models. So quite a lot of parameters, but we could, instead of thinking of some, like before, some structure for the overall matrix, we could instead infer the parameters with some finalization or regularization, which basically says that we expect these networks to be similar to each other. And we let the data, in some sense, tell us how similar they are, or how much regularization we need. That's a different way of saying it. Also, we may want to say that they are sparse, there are zeros. So we reduce, in some sense, the number of parameters, as well as making the networks more similar to each other by in regularized approaches for inference of parameters. Useful. I mean, compared to before, we now have matrices that depend on K. So we do learn environment-specific interactions. These 2 microbes interacting in saliva, but not in throat, and so on. Maybe the bit of disadvantage is that I would like to know, in some sense, these are the 2 networks that are these 2 networks are similar, and in what sense or how much, and so on. Whereas in this case, this is all done a posteriori. You essentially get 13 networks, 1 for throat, 1 for saliva, and so on. Then you can do a posteriori. You can say, okay, these have 50% of edges in common. This one 20%. You do a posteriori, right? You would maybe like to do it in an overall analysis. Okay, so that's what leads me to the method that we developed. So let's, in some sense, start from the beginning. What could be the generative process behind the data? The model I'm going to show is mimicking this process. So first of all, we have some network which we don't know and we would like to recover. So the nodes are the OTU, the microbes, and there are some interactions between them that we would like to find out. There is going to be some marginal model that basically modulates the abundances individually of each microbe. So there's the network and the marginal model, and those 2, in a generative kind of process, give us one sample. And this one sample has abundances for each OTU. So then you do the experiment 100 times and you get 100 realizations. Okay, this could be the case in saliva, one of the environments. Maybe it's true. I drew some other network, similar, slightly different. So there's quite a lot of sharing, and some are not, which then generates its data. Okay, so in a model I just showed to you before, essentially you do analysis independently, one for each condition. But how could we instead capture the fact that also in the generative process we expect similarities between these environments? Okay, the way I'm going to think about it and model this is in the form of a latent space. So latent in the sense that it's unknown, and again, we would like to find out from data. You can do it in different ways. You will see a lot the top part of this, in the sense that I see the body sites, the environments in a generic context, as placed in a 2D or 3D, up to us, space. So if two things are close to each other in this space, they will generate networks that are similar to each other, and then so on, the data. If they are far away, the two networks will be different, and so on, and the data. So this is part of, in some sense, of the generative process. There is an underlying latent space where these conditions are located, and the closer they are, the more similar the networks. And I would like to find out, in some sense, where these things belong, where they are located on this space. In theory, you could have a space also for the nodes, so where if they are close in this space, they're more likely to connect, for example. So, this idea is quite general, but as far as we are concerned, I guess the top part is what makes sense. So indeed, in general, what we talk about is a so-called random graphical model, which doesn't exist, or at least not before this paper, in the sense that you probably know a graphical model. Well, that's the bottom part, and that's the one I discussed before. So for each environment, you define a graphical model, Gaussian, Coppola, whatever. We need to decide depending on the data. This top part is in some sense new when you are in the field of graphical models. So we would like to define also a model that describes the distribution of the graphs. So each of these, there is a network, a graph, which has a statistical, it's a conditional independence graph in a graphical modeling literature. And so we have one for each condition, and we try to model this. How are they generated jointly? And we do this, we specify a model with some parameters, and hopefully these parameters will also tell us relationship between the graphs. And I call this random graph model because up here is essentially a modeling of networks. So it belongs to the field of network science, modeling of network data, where they call them random graph model, because it's the graph that is random. The random variables are the actual interactions, whereas down here the random variables are the nodes. So it's a bit of a combination between kind of modeling the graphs and modeling the stochastic processes on the nodes. And so we came up with this mix of random graphical model. Okay, so now this is quite generic. I will now go back to the microbiome in the sense that we need to specify this model in some sense and on some application, as well as this one. Okay, so therefore I will give you, I will tell you basically how we define the models for the microbiome data. Let me go one by one so you see how. So there's going to be one bit about how we define the random graph model, and the second bit about how we define the graphical models for each environment. And this essentially defines the full model with the parameters theta, omega, and so on, which will then need to be inferred from data. Okay, so the first one is this random graph model. Okay, we have a choice here. This is the one we chose, some kind of sensible, useful model, considering the type of questions that we have and the data that we have, essentially. So at this stage, you can think of 13, in our case, the environments networks, which let's see, someone gives us these networks. Eventually, they come from the graphical model. But let's say for now that we have these networks and we want to model the formation of edges in these networks. So the modeling point of view, I'm thinking, what is the probability that two nodes, I and j, are connected in environment K, right? Because if I'm modeling the graph, I'm thinking of probabilities of edges being there or not. And I want to do it jointly, but in fact, I do it conditional, okay? Conditional on everything else. So as I said, we have a choice here, and these are all parameters. This you see here is called the probit regression model. Maybe you're more familiar with logistic regression, would be okay too. So some kind of model where the responses are binary. So I put in this model various parameters. The first one is some intercept. Maybe there is more sparsity in one network than the other. So maybe saliva is very sparse and throat not. So I put a parameter that depends on K. The second bit, maybe we do have some data that tell us whether two nodes are more likely or not to connect. So some really regression type of model. So with some regression parameters beta, you will see an example later when we have those. So maybe you have some actual data that tells you whether the probability is higher for two nodes to connect or not. I suppose this is the main thing, this latent space business. So we give to each environment a vector. We can decide, I put it in R2, and this is the most common. You could say R3, then you have more parameters. So these are parameters. So each environment gets two parameters, which is the location, the coordinates of that environment on this latent space. Now, again, another choice. What is the distance between two of these points in this space? Up to us. We chose the inner product that you see here. But it's a choice again, so choices and like that. But the most important point, we write it like this. So if ij are connected in environment K prime, another one, then and if K prime is close to K in this latent space, this will increase the probability of also I and J being connected in K. Yeah? So it's this idea of if they are close in this latent environment, they are more likely to have similar networks. If they're distant, they're more likely to not be similar, and close or distant you define with some distance. We took the inner product and we took R2. These are all some such choices. Okay.
The variables are the C's. The C are the variables.
The parameters are the C's. Yeah, parameters. The variable, like the random variables, are the graphs, the edges. So the parameters are alpha K, beta, and all these CK. Yeah. Is that? Oh yeah. So sorry. Yeah. So this is about the graph in condition K, conditional on the other ones. Minus K, so you have saliva conditional on throat, all the other 12 body sites which appear here, because this is a sum for K prime different to K. Yeah. Okay. Okay, so that's the kind of first model. The other one, maybe you're more familiar with, some kind of graphical modeling. As I said, the data are not Gaussian, so we use Gaussian cupola. So I'll quickly maybe tell you a bit about this. So indeed, this is the notation I showed to you before. So it's this p-dimensional vector of microbial abundances in condition K. They will have some distribution. You can always, if I know F, I can always find a cupola such that this is true. So the cupola couples the marginals and the dependence structure. Obviously, I don't know F, but I then can go backwards in some sense. I choose a cupola, and so I model the joint distribution with that. And it's quite convenient to use a Gaussian cupola, which you see written here, because then you can, in some sense, go back to Gaussian graphical models. So you go back to this precision matrix, the inverse of this K, which gives you this dependent structure between the microbes, but accounting now for marginals that are not Gaussian. So if you haven't used Gaussian graphical models, I mean, you can look at this, in some sense, transformation here. So Y is the data, and F here are the marginal distribution of each microbe. So this you can, if the Y is continuous, this is a one-to-one transformation to Gaussian data on which you then do Gaussian graphical models. The data are not continuous, which makes it a little bit more difficult. But if you've never seen cupola graphical models, you can think of starting from your data, fitting marginals for each variable, and then transforming the data with this transformation into Gaussian, multivariate Gaussian data on which you do Gaussian graphical models. Okay, you do this for each case. So we want to have precision matrix that depend on the body site. Okay, so maybe this thing I was going to take one. Okay, yeah. So yeah, I will briefly tell you a few complications in some sense. I mean, those marginals, they need to be estimated from data. Typically, people will take non-parametric empirical CDFs from the data. But I mean, you could have other variables, for example, other data for the patient. You may know the age, the gender, all kind of metadata that sometimes is collected. And in that case, one could actually fit regression models to estimate the marginals. Okay, maybe I'll skip a bit, but you then would have to choose which parametric model you're going to use and so on. So we use a discrete wave, maybe not so common, negative binomial, if you know it, negative binomial regression, or even Poisson regression, to basically connect the marginals with external data that you may have and typically you have for this type of data. Okay, so I would like to put now, so hopefully you have an idea of this complex model, which puts together all the information from all the body sites. And now we, in some sense, so this mimics the generative process. So if I give you a theta, you would be able to generate graphs with this Bernoulli, and you would be able to then generate precision matrix, normal data, and with the marginals count, you would generate count data. But now we have to go backwards in some sense. Now we have the data, and from that, we need to recover the parameters. The parameters are possibly parameters in the marginals, okay? The last thing I said, but most importantly, parameters, those P by P, 87 by 87 precision matrices for each environment. And okay, those are the parameters, and the theta, those locations in the latent space, alpha, betas, these are all quite a lot of parameters that we would like to infer from data. Some complications, I'll go quickly so then I can show you the results and maybe it makes a bit more sense that way. So some complications, the data are discrete, they are not continuous. When you use cupola, this makes a complication in the sense that there is no one-to-one transformation between a discrete data and a continuous one. So in fact, the discrete data, as you can see here, is, I don't know, one, a count of one for one OTU at a certain probability mass function, certain probability here. It actually corresponds to an interval for the Gaussian random variable on which you do the Gaussian graphical model. And this interval, the kind of the red, the integral here, is the same as the probability here. So in other ways, the CDF have steps, right? And you need to account for this in the mapping between discrete and continuous data. So in fact, the data for the graphical model, it comes along in some sense in the form of intervals for each observation. In the literature, also on the statistical methodology literature, not just applied, they just condense this interval into a point often. Like you take the medium step in the CDF, and that's it, and you do the transformation with that. But I mean, you can do more advanced procedures, which is what we do by sampling from this interval. Essentially, when you observe in one, your statistical procedure will sample Z in this interval here. So that's the more accurate way of doing it. The other thing is this is the more complex in the sense of high dimensional. So you have graphs which are really 87 by 87 nodes. They could be even larger, in fact, and loads of parameters floating about. So you need to be able to search this space. We do it in a Bayesian way, which in some sense makes it more difficult. So you put priors on parameters, and we developed the Bayesian Markov chain Monte Carlo procedure to then sample from the posterior. I'm going quickly. I don't know. I mean, maybe people are not. I will give you references later if you want to check. I mean, the first bit is some pro-bit regression. There are Bayesian inference procedures for that. The second bit is Bayesian procedures for inference of Gaussian Coppola graphical models. There are papers also for that. Maybe you're familiar with that. In some sense, we connect the two. So when you are in an MCMC, when you are in the space of a graph and you would like to decide if to add an edge in a particular condition or not, for that, you need to consider all parameters, including this theta, this location in the latent space. So the two procedures obviously are all connected, right? So I decide, or the statistical procedure decides to add or remove an edge, also on the basis of the location of the body sites on the latent space. Okay, so why we do it Bayesian? Yeah, sometimes I also ask myself, typically I don't do things Bayesian. The nice thing, we did it here, but the nice thing is that it's this huge space of graphs, right? Imagine how many graphs you can draw for 87 nodes and 13 body sites. Huge space. So it's unlikely that you have a nice likelihood with a nice maximum, the things that we'll teach at our courses. So in some sense, Bayesian is more useful because it gives you, for example, posterior probability of two microbes to be connected in a particular environment. So it's not a 0-1, it's there or it's not there, this is the optimal model and this is great. It's more like accounting for uncertainty and variation, which is useful in this huge space. Okay, I would like to tell you a bit about the results. Hopefully then you see maybe how it could maybe useful, this way of modeling the data. And then if I have time, I'll tell you also another application. So this is one way of visualizing the results. So each column is one of the graphs. I visualize a graph as a vector. And this vector contains the posterior probabilities of each edge. So if you see blue, in fact, you see a lot of blue, these posterior probabilities are small, close to 0. So that's an indication of a lot of blue is an indication of a sparse graph. Whereas red is a posterior probability close to 1. So we are quite sure, or the procedure is quite sure that there is an edge there between these two microbes in that environment. So what we are supposed to see from this visualization of the results, first of all, there's a lot of blue, a lot of sparsity. That's typically what we expect. Second, if you go horizontal, you see some connections that are there for all the environments. So these are what we say the core network. These microbes interact in saliva, throat, and all these different environments. On the other hand, you see that there is also some, for example, these environments, on the x-axis you have environments. So these environments here tend to have quite a similar structure. So if an edge is present in all, and if it's not present, it's not present in all. And also these ones down here. So this is all captured at the level of this model at the of graph. So let's have a look at that. So that's the model that we fit. Okay, the alpha K are the sparsities. They are related to the blue and the red that you saw before, so I'm going to skip that. But we included some covariates. So, I mean, there are things that biologists know about the data or general information. For example, there is a taxonomy that tells you, that puts together microbes in the same taxonomy. Those are the names, phylum, class. We don't know much about that, but they do. And they think that if two microbes belong to the same taxonomy, they are more likely to be connected, to share some mechanisms. So we put that in the data, in the sense that there is data about which microbe belongs to which taxonomy. And you could say if they belong to the same taxonomy, 1, they are more likely to connect compared to not belonging to the same taxonomy, 0, in terms of here you see the betas. So on the right-hand side, you see the actual inference. It's Bayesian, so you see posterior, but if you just concentrate on the median, the mode of this distribution, you can see that for some of them, at least, it's positive. So indeed, also the data seems to suggest that if microbes belong to the same taxonomy, they are more likely to connect. More interestingly, I guess, considering all the build-up to this, is this latent space. Again, this is something that comes out from the inference. So from the location of these body sites on this 2D space is something that is learned by the model, by the inference, statistical procedure from data. Okay, you don't see much. This is subgingiva and supgingiva, if you could read it at the back. So these two, in fact, they are all very close. They have two networks that are very similar to each other. So indeed, this is what the procedure, the statistical inference seems to find out. Throat and tonsil, also in the mouth, are also have similar networks, but actually different from this one. I also put some colors to, they call it Oberon anatomy, so the actual location of the body site. So, for example, left and right ear are close to each other. So, I mean, there is nose is for some reason close to stool, but I think it's not what we are thinking about. So in any case, there is basically this is a result of the statistical procedure. So you can look at it, and you can also measure distances between the networks of the different body sites. Okay, so one more thing before I can show you another application if you have enough of microbes. So is this useful from a statistical point of view? I mean, can I not do, like I said at the beginning, can I not do a network? Yeah, maybe that's close. Yeah. Can I not just do a network for each body site, a graphical model for each body site, and then I look at the overlaps and so on a posteriori? That's one of the things I mentioned at the beginning. Yes, you can do this, but when there is a lot of similarities between networks, then there is much more. I mean, from a statistical point of view, it's much more powerful to put the data together. There's a lot of sharing between different body sites, which you can only account for if you model the data together. If you model it separately, you lose this sharing. So this is something we try to visualize here. On the y-axis, you see the variance, basically. For each edge, we take the posterior probability of that edge times 1 minus p. So basically, if it's 0.5, you're doing quite badly. So every edge, you're tossing a coin to decide if it's there or not. If it's close to 0 instead, it means that these probabilities are either 0 or 1, which means you're quite certain whether this edge is there or not. And you can see that if you do analysis for each condition separately, it's the red boxes. So you have a bit more variance in the results. Whereas if you do analysis with this method I presented, you gain in terms of accuracy and precision. Okay, so for the last part of the talk, the last 10 minutes or so, I can show you another application, just to maybe give you an idea, a more general idea, is this useful? So in general, what we are thinking about is data, multivariate data across different conditions. The microbes is one example, but you have plenty of other examples, and this was another study we did, where we, so now we don't have microbes, we have cultural traits. You will see what they are, and there's 10 of those. We don't have body sites, we have countries. Countries in the world. And there is plenty of data, survey data, World Value Survey and European Value Survey, where you ask people, again, different people for different countries. You cannot have the same person across all countries, obviously. So you take a pool of sample of people from different countries and you ask a number of questions, particularly four. What is your justification of homosexuality from 0 to 10? What is your, from 1 to 10? What is your justification of abortion from 1 to 10? How happy are you from 1 to 4? These kind of questions, right? So we take these 10 in particular because they are used in the literature. You can, in some sense, you can use these questions to define the culture of a nation in some abstract way. Obviously, the culture has got lots of dimensions. We take these 10 to represent the culture of a nation. And once again, like before with microbes, you could expect that, for example, if you are tolerant about homosexuality, maybe you're tolerant also about abortion. So there are some dependencies between some of these questions here that may be the case for all countries. So this could be the case of the core network between microbes. But perhaps some other questions or relationships between questions may be different for different countries. Maybe there are more religious countries, less religious ones, the importance of God and so on. So both at the marginal level, but also at the level of dependencies, you can have differences between countries. And so that's what we, in some sense, was the same idea that we used for microbes, we used it also here. Can we learn the network between these dimensions of culture? Can we see how different it is between countries? So now you know everything about the model, so I don't tell you again about the model, but this is immediately the output, in particular this latent space. You can't read anything, and it's okay, but at least I should tell you what each of these location now is a country. So you can't read, but for example, down here, it's France and Germany. So they are close to each other. What does it mean? Well, it means that the networks are similar to each other, and so in a more abstract sense, that the culture of these nations is more similar, which, okay, I mean, this is about the world. So, I mean, France and Germany are similar to each other if you think about the world. The specificities that are not like... and so on. So, but the nice, I mean, what you can see here is a big spread, right? Which in statistical terms, it's a large heterogeneity. There is quite a lot of difference between countries. So this is a bit the end. This was, in some sense, it was the end of the last example on microbes. But I'm showing you this because we could go further in some sense. So you see this plot, you see a lot of heterogeneity, and you think, okay, can I explain this heterogeneity in some way? I mean, from a statistical modeler perspective, you want to explain what you see. You're not happy just to see a lot of variability. Of course, this tells you more. It's not just variability, it also tells you what is close to each other and what is far away. But can I actually explain it? In this example, it's somewhat simpler because we don't need to know biology. We can get to there by ourselves. Okay, France and Germany close to each other. Geographically, they're also close to each other. So perhaps distance between countries can explain some of this variability. Okay, fine, but we can measure it and we can put it in the model, right? So this is, again, don't worry too much about the names. There is still some France and Germany down here. So here you see lots of colors. I mean, blue is Europe. So what you're supposed to see is that indeed Italy is here. So countries close to each other geographically, they also have similar culture and therefore similar networks. Whereas, I don't know, the more pink ones are Asia. Up here in green is Africa. So you do tend to see some how geographical distance explains a bit the heterogeneity. And as I said, we can just, I'll show you later. You can just put it in the model. Okay, the next one. I mean, distance doesn't explain everything. You can't read it, but you can trust me. This is New Zealand, and this one is US. They're quite far geographically, but they seem to be quite close in this latent space. Okay, but they speak the same language, but more importantly, they share a history, right, of colonization, which may have then ended up in the same also cultural values. But again, we know this, we can put this in some sense into the model. So this is a more refined version with just the 5 main languages. I just take the countries that speak these 5 main languages, and in fact, you can see now, for example, New Zealand and the US here. So indeed, they are close to each other because of this shared history. And as I said, we can include this in the model. So now the model becomes more complex. Before we had this latent space. Now in some sense, we put some measured distances, some kind of measure of similarity. So 1 over the distance, for example, that tells us that again, if 2 countries are close to each other geographically, we expect the networks to be more similar to each other. And then we let the data tell us, is there this effect or not? And yes, the data now down here does indeed tell us that spatial proximity is quite a strong effect, and also language. Yeah.
So in the previous slides, isn't it an effect of projection? Their projection are close.
They look, yeah, I mean, you mean, yeah, you can rotate them. Yeah, so you just need to look at them relative to each other. You can rotate this, and the inference will be invariant to that. Yeah, that's a good point. Yeah. So it's really just about relative kind of distances between 2 countries. Yeah, so indeed you see geographical distance having a strong effect, and also language, primary language spoken, having a strong effect. And the nice thing of doing this is that if you start to think carefully about what to include in the model, in some sense, this latent space loses importance. And that's good in some sense. You would like to explain that heterogeneity as well as you can with data and models, so that the residual, so what is left over, is just noise, and there's no structure, which you kind of see here. Again, don't worry about the actual names, but what you should see is lots of heterogeneity on the left, which indeed is when we did not include these covariates. When you include them, then there is very little structure left in the latent space. And if you do model selection, you would not select anymore this model with so many parameters and latent space and so on. You've done a good job. In some sense, you put a lot of interesting variables in the model so that this what is left is just noise. And that's how it should be, and it's useful. Okay, so yeah, so this time is up. If you hope you followed, but if you want to know more, those are the two papers: the microbiome on top and the national cultures at the bottom. We also have a package if you use R, so you can actually try this. And yeah, this is a brief summary of trying to do this modeling of multivariate data for different conditions. So you have this Gaussian coprolographical model for each environment. And I suppose the more, let's say, novel thing is this model of the graphs on top of that, which also explains relationships between the graphs and the network. Okay, I'll stop here. Okay.
Oh, nice talk. Other questions?
Yeah.
Can you explain a bit again the previous slide? These two graphs.
Yeah, so yeah, maybe I can. So on the top here, you see a very simple model where there is just an intercept and this latent space. So then you see how the latent space is essentially, besides the intercept, is going to capture everything else in the model, all the similarities and differences between the networks. So in the later on, we tried more refined model where you see the blue part here, where you put more variables, observed variables, because we have measurements of distance, geographical distance, whether they speak the same language. We put that into the model to explain whether I and J are connected in K and more likely to be connected if they are connected in a country that is close geographically, or if they are connected in a country that speaks the same language. We include that into the model. So then this part here, in some sense, if you think of a linear regression, is not, but you can think in the same way. That part goes, is what is explains what is left once you account for these covariates. So if these covariates are really important, what is left will have less structure. They will account for a lot of the structure that you see, which is supposed to be shown then here. So when you put interesting variables in, so the rest is all around zero and not just noise. Yeah. It's the plot of so on the right here you have the plot of the latent part of this model here. Yeah.
The CK.
Yeah.
And you choose two dimensional.
We choose two-dimensional, which this model, not in the combination with the random graphical model, but this model is in the literature. It's called the latent probit model. Sometimes they use it with three, but then you have three parameters for each body site, so many more. And you don't have nice visualizations. I mean, you have 3D visualizations. But yeah, I mean, you can do it. It's essentially more parameters to fit. But yeah.
Why should you also?
Yeah, two is really pure. Two is purely for visualization. So it's a 2D space rather than 3D, and parameters. So you include one more dimension, you will have 13 more parameters. Not that many, but okay, actually countries we said 87, so 87 more parameters. Depends in some sense how much data you have, but the more formal procedure will look at this also as a parameter to select. So you try 2, you try 3, you try 4, and then you see what the data tells you. Okay, given how much data you have, you should go for 2 or for 3. It's just, you wait much longer, and but yeah.
So,
When you were showing your random charts, were you showing it as adjacency matrices?
mean in the code or in the? I mean, in the slides, I never really store them too much, in the sense that here you see the entry ij. So yeah, ij, I suppose that's an adjacency matrix. Yeah, but when I was doing plots like this one, then I look at them as vectors, so the upper triangular matrix of this adjacency matrix. Microbes. No, actually, I should say that it's the same microbe. So the experiments will look at, in fact, many more, 10,000 of those. So simultaneously, one experiment gives you the abundances of all microbes. So yes, for the analysis, I should say, you need the same variables across the different environments, which in fact for survey is a bit of an issue. Differently to what you think. Instead, there is quite a bit. Maybe one question is answered in one country, but I don't know, justification for homosexuality in some countries will not allow that question to be there. So actually, no, there is more. There is more filtering to do in the survey data. In some sense, maybe some years have these questions, but some years don't have it. So you actually need data for the same questions across countries.
yeah, so this way of inferring data is very flexible, as far as I understand. And then you read the relations between your data from the graph, and that gives you like a first order of relationship between the data. So is there a way to, distinguish, how the relations are important, the importance of these relations?
Or yeah,
like first order, second order, you know.
I don't know what you mean by order, but in terms of yeah.
When let me say, in the example that you've done, you say these countries are very close to each other, but then they are very also close in terms of distance, and then you infer that.
Yeah.
And then you say, okay, but then there was the colonization. So, I mean, in your human way of thinking, you have a certain order of importance of this relation, right? Can you read this from?
I don't know if I understand the correct meaning of order, but I mean, I can try to say something and then maybe it's, you see if it's useful. First of all, there are these parameters that are one for each of these dimensions. So whether it's language or geography, they each get their own beta, which in some sense that tells you how much and in what way they influence the edge. I don't know if that answers your question. The other thing I could say is that I showed here, for example, each edge gets a probability. So you have a range between 0 and 1, and you can say, okay, this is this edge is strongly in the graph and not. So some ordering in that sense. But that's only one measure. For example, I could also look at the omega, the partial correlations, let's say, that tells me how much these two are associated positively, negatively, blah, blah, in one environment rather than another. So this, if that was your question, I mean, this latent space is just about the structure of the graphs. For example, I could go a bit more precise and I could say, actually, if they are positively correlated in one body site and they're positively correlated in another one, then they're close to each other, not just whether it's an edge or not. But I don't know, I feel I haven't fully answered your question, but maybe we can discuss later. These are all things that are related to. Yeah.
It seems that you have some papers on like causal modeling with mixed graphs and like similarly vertical graphs and stuff. Could you maybe comment on like where you see some benefits from the causal modeling in the microbiome?
Yeah. So I couldn't connect it to tensors, but that is my my main research expertise at the moment, causality. So yeah, I mean, it's quite far in the sense that here it's all about undirected graphs. So I'm looking at directions between cultural traits and so on. I suppose, I mean, it's already difficult to reconstruct a causal graph from just one condition, one environment. I don't think that the research has fully answered that question. That's what I'm interested in. So is this variable having a causal effect on another one? We are not fully there yet. So once we get there, then this in some sense comes after. Okay, then I have different environments and I have a causal graph for each environment and I want to learn how similar they are.
Experience so far, has there been like a difference between trying to learn the maximally ancestral graphs versus like compared to the undirected graphs?
Oh yeah. Oh yeah, loads of difference. Yeah, I mean, this is more at the level of regression modeling, let's say. So causality is quite a different thing. So now I guess I don't know how much the others are familiar with because you're saying ancestral graphs. So if you have the parents of a node and then the children of the node, for example, just a simple causal graph. The children are usually the annoying, they're always annoying, but I mean, in this particular case, they're quite annoying. They're basically very much predictive of of the nodes that you're trying to predict, but they are causes of this node. So in a regression type of analysis, you can't do anything about the children. They will be there and they will be connected. In a causal analysis, precisely the ones that you want to exclude because you want to just have the causes. So yeah, no. So
in terms of the statistical performance, does it help or how much does it help? Right, we've seen these examples of like the sort of regression analysis, but when it's a causal.
Yeah, I mean, if you are just interested in correlations.
10%
Yeah, and sometimes the question is different. If you're just interested in associations, predictive dependencies, then regression is enough. If you're interested in causal mechanisms, what causes what, then causality is.
In terms of the statistical performance for the prediction, it doesn't help.
The prediction, no, you should in-sample prediction at least. I mean, if you then do machine learning and want to predict out of the sample that you have, you need causality again. Okay, well, maybe this gets us way too far. So I think we can, I'm staying here for the coffee. So if anyone has questions.
Okay, so the same for